Today, we are going to study how to memorize amino acid structures. All of you know that there are 20 standard amino acids which occur in almost all organisms, including archaea, eubacteria, and higher organisms. Why there are only 20 standard amino acids? Have you ever thought of this aspect? Why there could be only 20 amino acids? Of course, there are a lot of other amino acids such as citrulline, but they are not seen in the structure of proteins. Why? The answer is these amino acids, these 20 amino acids, do not cause much steric hindrance in protein structure, so that the protein conformations remain stable. Other amino acids, non-standard amino acids, have a little complicated structures so that they cannot be easily accommodated into the protein structure. So these 20 amino acids are called standard amino acids or canonical amino acids. Now let us see how to memorize the structures of all these 20 standard amino acids. Many of you may be not interested to study the structures, but this is actually very easy once you practice. Once you know the essence, it is very easy. So give it a try. So first you have to understand the general structure of amino acids. Here you can see there is a central alpha carbon which has valency of 4. It can make 4 covalent bonds. So to the right side, as in the picture I am seeing, to the right side, it is connected to a carboxyl group COOH. On the left side, it is connected to an amino group NH2. On the downside, it is connected to a hydrogen. On the upper side, it is connected to an R group. It can be the R group can be a simple methyl group, or it can be ring structures, or it can be aliphatic long chain molecules that we are going to see in further slides. So this basic structure you have to study to understand the structures of all the 20 amino acids. This will help you to memorize, because the only thing that changes in the structure of an amino acid is the R group. So now let us see how the R groups are changing in different amino acids. So which is the simplest amino acid? The simplest amino acid is glycine. Now let us see the structure of glycine. So this is the structure of glycine. So can you see what is the molecule that gives the identity to glycine? See the hydrogen. See, this hydrogen is the R group in glycine is a simple moiety of hydrogen, right? So this is the simplest amino acid. Now let us see the next one, Alanin. So what is the change here when compared with glycine? You see, so it is the methyl group. The H in the glycine has been replaced by a methyl group. So this is the identity of alanine, right? So this is how it is very easy to study the structures of amino acids. Now you can see that alanine is a glycine derivative. Only one molecule is changed. Now let us see valine. So here what is the change? In Aladdin, it was a methyl group, so it was CH3. Now here you can see the CH3 has been changed and two hydrogens from the CH3 has been removed and replaced by two additional methyl groups. So you can see here in this ring two additional methyl groups. So this is how alanine becomes valine. So valine is an alanine derivative. Most of the amino acids are actually the derivatives of alanine. You will understand it further. Now let us see leucine. So what is the change here? See that CH3 has been replaced. You see the valine structure is still there, but you can see an extended CS2. See, right? So it's very easy. Now let us see isoleucine. See this. One methyl group is there. So basically that CH3 of alanine is still there. See. So here the one chain gets extended little further. Now methionine. Methionine is a sulfur containing amino acid. So there are only two sulfur containing amino acids among the standard amino acids, methionine and cysteine. Now let us see the structure. What makes the change? You see this. Alanine had CH3. So one hydrogen is changed and a chain of sulfur containing side chain is attached. So that is a CH2S CH3 chain. So this is methionine. Now sulfur is present in between these two carbon molecules. Now another sulfur containing amino acid is cysteine. Here you can see it has a sulfhydryl group. So it is basically a methyl group in which one hydrogen has been replaced by a sulfhydryl group. Next is serine. Serine and threonine are two hydroxyl group containing amino acids. So here you can see the hydroxyl group. See, but you can see that the methyl group is still there. CH2 is there. The CH2, one hydrogen has been replaced with OH. So this is also an alanine derivative only, right? Next is threonine. So here what happens? So this also has the methyl group initially, but two of the hydrogens have been replaced and with one hydroxyl group and one methyl group. See this. So this is also an alanine derivative, right? Now coming to aspartate. Aspartate has it is having one additional carboxyl group, that is it is usually called aspartic acid, right? So this is also a methyl group derivative. How? See, the CH3 has lost one hydrogen and replaced with one carboxyl group. You see COOH, right? Now glutamic acid or glutamate. So this is another acidic one which has, you see this is also alanine derivative. How? You see that this is with an extended CH2 chain here. You see this is the extended, otherwise it is aspartate only. So when this CH2 comes, it becomes glutamate. So this is how you can easily remember the structure of aspartate and connect it with the structure of glutamate, right? Now coming to asparagine. Hmm? Asparagine is an amide of aspartate. This is the definition. So what is an amide? You can see here. So it is an amide of aspartate. So you can see on the left side, you can see the structure of asparagine. On the right side, I have given the structure of asparagine, and on the left side, I have given the structure of aspartate, and on the right side, I have given the structure of asparagine. Right? You see here. So this amino group So that OH group of the aspartate has been replaced with an NS2. You see this OH of aspartate is replaced with an amino group. So it has an extra amino group. Hence it is called as amide of aspartate. So if you know the structure of aspartate, it is easy to know the structure, easy to memorize the structure of aspartate. It's all connected. It's all connected to alanine. You can still see that alanine derivative thing here. Now, glutamine. Glutamine is an amide of glutamate. Glutamine is an amide of glutamate. So if you know the structure of glutamate, that is also an alanine derivative, you can only what you have to do is only add an NS2 by replacing an OH. So this, when hydroxyl group is replaced by an amino group, the glutamate becomes glutamine. Now lysine. This is a positively charged amino acid, usually seen in histone proteins and all. Now here, this one also, you can see that there is a long chain of uh, CS2, 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 then followed with one amino group which has a positive charge that makes it positive. Okay. Now, arginine, this is another positively charged amino acids. Here also there is a long chain of CS2, CS2, CS2 followed with one NH, then a carbon which is further connected to two more molecules of NH, of which one has a positive charge. So this also is a positively charged amino acid and hence found in the histone protein associated with the DNA molecules. Now, histidine. This is also a positively charged amino acid. You can see that here in this ring, which is called as this is a pentagonal ring. This is also an alanine derivative. You can see the CS2 there, and to the CS2 is connected one pentagonal this ring, which is called as an imidazole ring. So of the CS3 methyl group, one hydrogen has been replaced with an imidazole ring. Okay. So next is proline. This is the most structurally the most complicated amino acid. See, proline. Proline has, if you carefully observe, the methyl group is forming a ring, sorry, a chain of CS2, 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 and then getting it connected to the amino group of the parent chain. So this process is called internal cyclization. Okay, you see, I will show you further. You see, this is internal cyclization. So the chain in the same molecule become attached with the same skeletal structure is called internal cyclization. You can see how it happens. See, this is how. See, this turns and gets connected to the amino group. Right? You got it. So this is the most structurally complicated amino acid because of the internal cyclization. Next is aromatic amino acids. This is phenylalanine. You can see this is also an alanine derivative. You see CS3, out of the CS3, one hydrogen is replaced with a ring, a phenyl ring. You see this. So this is very easy to memorize. This is a hexagonal ring only, right? This is tyrosine. You see, this is also an alanine derivative. You can see that methyl group there. One hydrogen is being replaced with a phenyl group, but there is a change. See this OH. There is an OH attached to the phenyl group. Hence, this minor change makes it different from phenylalanine and tyrosine, right? Now this is the tryptophan molecule. Here you can see Still methyl group is there. See, that this is also alanine derivative only. But the methyl group has lost one hydrogen and one double ring structure has been attached to it. So this double ring is called, you see this is the double ring structure. This double ring is called indole ring. Okay. So this is the only amino acid which has a double ring structure, and this is the what uh, the plant hormone called auxin is synthesized from this amino acid. This is the precursor of tryptophan, sorry, auxin biosynthesis. So this is how you have to understand the structural changes in amino acids, and that makes you memorize all the 20 amino acid structures very easily. So you try this and memorize it, right? You try to memorize, then it will be very easy to memorize the structures of 20 standard amino acids. So be cool and understand these simple structural differences. Memorizing becomes easy. Thank you very much.